So in this lecture, we will talk about uh, what type of assays people use to measure mRNA function. Um, so far, we discussed about how to design therapeutic mRNA sequences, what features matter, what trade-offs exist, how clinical goals change the optimization problem. But uh, design decisions only make sense if we can actually measure what those sequences do. Every sequence function model depends on experimental data, expression levels, and decay rates, immune system activation, stability. All of these come from biological assays. So when we build predictive models for therapeutic mRNA, so the model doesn't see, actual biology directly, right? Obviously, it sees a measurements we provide, and those measurements come from specific experimental systems, a particular cell type or a particular delivery method, a time point, or what kind of readout is it. So what we call ground truth is not universal biological truth. It is more, assay defined truth. Different assay systems capture different parts of biology. So understanding how those measurements are generated is essential before you interpret or trust any predictive model. And so all experimental systems sit on a fundamental trade-off between scale and biological fidelity. So scale means how many sequence variants we can measure, and biological fidelity means how completely the system captures what actually happens in vivo. Um, so when it comes to high throughput systems, they can test thousands or even millions of sequences, but they often simplify biology. So they may not capture physiologic RNA decay, innate immune sensing, intracellular trafficking, cell type specific translation controls. So more realistic systems include those processes, but obviously they are slower, much more expensive, and can test far fewer sequences. So this trade-off directly determines how informative our measurements are. The quality of model depends on the biological fidelity of the labels they were trained on. Here we can see how experimental systems differ in what they measure. All assays operate along these trade-offs between throughput and biological practicality. So when it comes to cell-free systems, they can test large numbers of sequences quickly, but they capture only core translation mechanics. They lack compartmentalization, immune sensing, and many native decay process generally. So whereas cell-based systems introduce, real cellular machinery, including RNA turnover and some innate sensing, but still miss organism level biology, obviously. So in vivo systems provide the most complete picture, immune activation, pharmacokinetics, tissue targeting, whole organism physiology, to name a few. But they are low throughput because no single system captures everything. So ground truth is not measured in one place. So it's built by integrating data. So here let's see how labels are generated. And then we already learned that models don't really observe biology directly. So they learn from our measurements, and different measurement systems capture different aspects of function. At the highest scale, we use these high throughput screens. So things like mRNA or large reporter assays. So these let us test enormous numbers of sequences in parallel. They are ideal for generating training data, but um, they measure simplified biology. And the next we have mechanistic assays. So these isolate specific processes, how efficiently ribosomes load, how fast RNA decays, but they don't scale as easily, but they provide more direct measurements of contributing mechanisms. And finally, so there are physiological assays. These measure what ultimately matters in a living organism. Protein output, tissue behavior, systemic effects. So they capture full biological context, but they are low throughput and expensive. So functional labels are not all the same, right? So they depend entirely on which measurement system produced them and which layer of biology that system can see. So let's start with cell-free systems. First, let's start with the strengths. So because they remove cellular complexity, they are easy to scale. You can test massive numbers of sequence variants quickly and at relatively low cost. They also produce, quantitative signals across a wide measurement range. So that makes them well suited for comparing large libraries of sequences. Codon changes or structural elements, motif variants, for example. So most importantly, they let us explore sequence space broadly, you know. So they're not about capturing full biology. They are about mapping functional response across many variants. So that's why, cell-free platforms, dominate discovery scale screening and large training data set generation. Now, let's talk about limitations. Yes, cell-free systems are powerful, but they remove a large portion of real cellular biology. First, key biological pathways are missing. There is no innate immune sensing, and RNA decay mechanisms, for example, are simplified. Um, when you compare with living cells, so that means some sequence behaviors that matter in vivo simply cannot be observed. And then the translation environment is fundamentally different. So ribosome loading, scanning and elongation occur without normal cellular regulatory control. And because there is no compartmentalization, spatial organization of translation and RNA turnover is lost. And then cell-free assays can introduce artifacts so that are not biologically meaningful. For example, cell-free systems often fail to show the inhibitory effects of double-strand RNA that would be seen in vivo. So it means cell-free measurements, reflect more of the assay environment, the physics of that, not necessarily the full biology of living cells. I mean, we're not saying, these systems are wrong, but it means their results must be interpreted within the context of what the system can and cannot represent. So, when it comes to cell-based systems, so they restore much of the biological context that's missing in cell-free systems. So RNA is processed within intact cellular machinery. Natural decay pathways operate. Translation initiation occurs through normal cap-dependent scanning, and elongation is regulated by the full cellular factors. Importantly, the spatial organization of the cell is preserved. RNA localization, compartmentalized translation, and regulated turnover. All of these, contribute to transcript behavior. So, and then, cell-based systems also introduce immune sensing, but um, this is actually model dependent. Different cell lines express very different levels of RNA sensors, like TLR7, 8, or interferon responsive proteins. So immune related measurements generally reflect the specific biology of the chosen model system. So because they capture real cellular regulation, cell-based assays provide a much stronger surrogate for in vivo expression and stability than cell-free systems. But I mean, they still simplify whole organism biology, but they represent a major step up in biological fidelity. So when it comes to limitations of these cell-based systems, yeah, I mean they provide greater biological reality, right? Sure, but that realism introduces important limitations. Experimental scale is reduced, and throughput is far lower than cell-free systems, and then so which limits how many sequence variants can be tested. And then also these measurements become cell state dependent. That is, their metabolic state, stress levels and health. So all these things influence gene expression. So even small differences in transfection efficiency can change measured output. So this introduces lot of biological variability that's not directly related to sequence. And experiments become highly context dependent. Batch effects often reflect genuine changes in cellular state. So because of all this, right, measured labels depend not only on the RNA sequence but also on the experimental environment. Measurement dynamic range is also narrower, so I mean all these things, all these factors make cellular measurements more biologically meaningful, all right, but uh, also more complex, and interpreting and then modeling also is a bit hard. Okay, so now when it comes to in vivo systems, they measure therapeutic performance at the level that ultimately matters, that is the whole organism. So they capture, pharmacokinetics and dynamics, and including how the RNA distributes across tissues, how long it persists, and how much functional protein is ultimately produced. So they also capture integrated immune responses, right, both the innate and adaptive, which strongly influence stability, translation, and clearance. So because all biological processes are present, in vivo measurements reflect the net outcome of delivery, cellular uptake, translation, degradation, and systemic regulation. However, so these experiments are extremely resource intensive. Throughput is very low, timelines are long, and there is inter-animal, physiological differences. They also introduce variability. As a result, in vivo data, cannot be generated at the scale needed for model training. Instead, it serves a different role, that is basically validation. So it tells us whether predictions made from higher throughput systems actually translate into organism level therapeutic performance. So overall, therapeutic mRNA development doesn't really depend on one experimental system. Self-free systems are used for large-scale exploration. So they give us a rapid scanning of sequence space and reveal intrinsic sequence level effects. So the goal here is, basically coverage. And then selected candidates are evaluated in cell-based systems. So here, these transcripts experience, a really realistic cellular process. Um, and then finally, top candidates are evaluated in vivo. So this confirms therapeutic performance at the organism level. So effective pipelines integrate all three of these systems to assemble trustworthy ground truth. So to summarize this lecture, fundamentally what we need to understand is that assays don't simply measure biology and just generate data, but they shape what the models can learn.